My name is Elayne Benson, and my project has been over how IL-32 overexpression is driven by DNA hypomethylation, contributing to an extracellular matrix remodeling phenotype in breast cancer stem cells. So a mammary breast cancer cell has varying developmental stages that it goes through. At each of these, which a mutation occurs, depends on the severity and aggression of the breast cancer itself. So when you have a mutation or cancer-causing issue in the stem cell specifics, if it is not properly targeted and treated, you have this increase in cancer stem cell expansion as well as relapse and metastasis. However, when you have this targeting of the cancer stem cells themselves and identification, you're able to see a more regression in these cases. So in order to look at the cancer stem cell specific developmental changes, we took an epigenetic approach to this. So epigenetics is a developmental change that occurs on the DNA itself without changing the DNA code. So these are going to be Histone Modifications, which is acetylation that occurs on lysine residues, as you can see in this picture, in the nucleosomes, the histones themselves, as well as methylation that can occur on DNA. And this is where we took the first look at things with methylation. So methylation occurs on what is known as CpG islands. So this is on your DNA where you have cytosine next to guanine specifically. This is the only place that methylation can occur, so cytosines are able to be methylated next to guanines, causing a decrease in expression. So when there is methylation that occurs, it's what's known as hypermethylation, and you have a decrease in expression. However, when you have non-CpG islands, so genes that do not contain these CpG sites, you do not have methylation occur, which would be hypomethylation, which leads to an increase in expression. So what we want to do is look to see that there is a difference between cancer stem cell specific breast cancers versus normal breast cancer cases. So what we did first is we did a 450K DNA methylation analysis to show what population or what specific cells have large populations of cancer stem cells. So what we use to look for that is what are known as EpCAM and CD49F markers. So these are epithelial specific differentiation markers. So when these are present, there is differentiation that occurs. However, we know that stem cells do not have this differentiation, especially for cancers, so they do not differentiate. As you can see from that first picture I showed you, there was a differentiation flow. However, when you have that cancer that occurs in the stem cell specifically, the differentiation does not occur. So we wanted to find models that have large populations of this cancer stem cell to figure out how we can better target these specific cells to decrease the aggression of them. So what we did is, as you can see, there's a large population in MCF10A with a p53 delete as well as p10 delete, which mimics mesenchymal characteristics of basal-like breast cancer, which have metastatic and invasive properties. So they have a large population of these negative EpCAM and CD49F markers. Next, we wanted to find a cell line that naturally mimicked these characteristics. So using MCF10A, which is a non-tumor causing breast cancer cell line, we did a p53 delete, which is a tumor suppressor gene, as well as a p10 delete, which deactivates the PI3-AKT pathway, causing there to be a constant activation of this pathway. And then we also did a double delete of those two on this MCF10A cell line. So what we saw is when we did these single deletes in the cell line, MCF10 cell line by itself, it showed to be normal-like, and they did not have large populations of this EpCAM negative CD49F subpopulation of cells. However, when we did the double delete of them, this shows to have basal-like properties, which would be your metastatic and invasive migratory breast cancer. They had large subpopulations of these EpCAM negative CD49F negative markers. So with that in mind, we needed to find a model that mimics that of p53 delete and p10 delete properties in a cell line. So we took multiple cell lines that were already established and known, and we did, using flow cytometry, we're able to sort the amount of cells that have these EpCAM negative, CD49F negative markers. So the cells that do have these two markers, we are classifying them as cancer stem cells. So when they have a high subpopulation of these cells with these specific negative markers, these cell lines are considered cancer stem cell rich. So using that information, we decided to use Cell 159 as our model, as you can see at the top of the second section of this table. And that is what we have used for experimentation as our experimentation to follow. So we have these cancer stem cell rich versus cancer stem cell poor cell lines that we see here using our EpCAM negative, CD49F negative markers. And since we are taking an epigenetic approach, we needed to see that there was a difference in methylation patterns of these cell lines. So using PCA of cancer stem cell rich and cancer stem cell poor lines, we were able to look at CpG island promoters as well as non-CpG island promoters. So as you can see, all these red dots representing each of the cell lines that we saw previously, that the cancer stem cell rich, all of them have very similar CpG islands, and non-CpG islands, which also are different from our cancer stem cell poor cell lines. So what we're looking at is we're trying to find a difference in the methylation patterns of these cell lines to show that there is a developmental difference in the DNA of cancer stem cell rich versus cancer stem cell poor cells. So with that, we did differential DNA methylation of these rich versus poor breast cancer cell lines, looking at CpG island promoters. So as you can see, the right side of this heat map is going to all the red looking blocks. Those are going to represent your cancer stem cell rich cell lines. And the rest of them are going to be your normal like cancer stem cell poor cell lines. And as you can see, there's a complete opposite effect on CpG island promoters when probed for, and you see the same pattern in non-CpG islands. So this is showing that there is a very large difference in the methylation patterns of these two specific cell lines having a rich population of cancer stem cells versus a poor population in breast cancer specifically. So next we looked at the CpG island promoters that were present in the cancer stem cell rich cell line populations, and we did a gene ontology analysis looking at the biological processes that are occurring in these. So these are going to be your hypermethylated areas of the cells. So what you're seeing is a decrease in expression of cell differentiation and developmental processes, which is common to see and what we expected, considering when you have cancer stem cell rich breast cancers, you're not having a large amount of differentiation. Your cells are being almost stuck in the stem cell process, and it's not able to produce the necessary cells to develop as an organism basis. So next we looked at non-CpG island promoters using the same processes, and we see that the hypomethylation causing an increase in expression was seen for inflammatory responses, your defense response, pretty much what we expected of an increase in your immune system defenses. However, it's known that in cancers you have an increase in your immune response and defense systems. However, they're not necessarily doing their job. The cancer is still present and it's not being attacked by itself, which is where treatments come in. So we want to figure out, of these genes that are being overexpressed but not doing their job, what are they doing and how can we target them to make them do their job in theory? So we looked at finding a specific gene that was hypomethylated, so increased expression in our cancer stem cell rich, and hypermethylated, causing a decrease in expression in our cancer stem cell pores. So what we see is we found IL-32 promoter to be hypomethylated in these cancer stem cell rich cell lines compared to the poor, and this was also validated by looking at the IL-32 expression when we altered NCF10A cell lines to have an increase in EpCAM negative CD49F negative cells, which would be an increase in cancer stem cells. So we see that we have this genetic change happening in IL-32 when you compare these two cells, but we needed to see that there was actually something occurring in the cancer and it being affected to cause it to be more severe, because these cancer stem cell rich cancers are known to have a greater increase in severity and aggression. So using the Cancer Genome Atlas, which is just a whole genome profiling of cancer patients. So we're able to pull out specific types of cancers, subtypes of these cancers, and looking at expression levels of genes of interest. So what we did is we used ER positive versus ER negative and looked at the methylation patterns of IL-32 as well as the IL-32 expression. What we found is ER negative had a greater increase in both the methylation and expression patterns compared to the ER positive. So your ER negative is going to be more common in your cancer stem cell rich, which would be your aggressive types of cancers. And when we looked at just overall the different subtypes of breast cancer, you see that compared to normal, luminal A, luminal B, and HER2, basal, which is your aggressive metastatic invasive breast cancer, has a very large amount of IL-32 methylation and IL-32 expression overall. So we see that this is actually causing more severity, and it correlates to the aggression of the cancer patients themselves. So we wanted to look at how we can look at the function of IL-32 and maybe alter it to potentially help improve the aggression and prognosis of our breast cancers. So previous literature has shown that IL-32 is a bromodomain recruiter. So bromodomains are used to locate histone acetylation that occurs, which is another form of epigenetics. So what we looked at doing was, if we can inhibit IL-32 related functions, can we also inhibit IL-32, and how is that going to affect the cancer and tumor itself? So when we treated our SUM159 cancer stem cell rich cells with JQ1, which is a known bromodomain inhibitor, we see after 8 hours and continuously after a very large significant decrease in IL-32 expression. So we see that there's a decrease in expression. However, we were unsure if that affected the tumor in any sense. So using the same cell lines with and without JQ1, these SUM159 cells, we used a cam-chick embryo xenograft model. And we were able to inject these cells with and without the treatment to see what the tumor does itself. So what it showed us that there was a suppression in the tumor growth as well as vascularization. So not only did this tumor shrink in size, it did not have as much blood flow, giving it the ability for growth. So overall, the inhibition of IL-32 related functions did cause a positive effect on the tumor physically as well as genetically. So next we want to see what happens to the rest of the cell and all the genes if we knock down this IL-32 specific gene. So using siRNA, we were able to knock down the IL-32 gene, as you can see from our Western blot on the left, and RNA sequencing was done on these two samples of a control and an siIL-32. What it showed was that with the knockdown of IL-32, you see an increase in extracellular matrix associated genes. So this is going to be collagen, laminin, integrin, so forth genes that produce these proteins. We also performed a phosphorylation array that showed that with this knockdown, there was an increase in ECM-related pathways. So not only do we have these genes being produced, the pathways that are associated with these genes are also being produced. So we have next our theory being that with this knockdown, this IL-32 itself is causing a remodeling of the extracellular matrix. So with that, we know these cells that have increased IL-32, these cancer stem cell rich, have an increased invasion compared to normal, like breast cancers. So we performed an extracellular matrix Transwell invasion assay. So with this, you can see that our control compared to our knockdown of this IL-32 gene showed a significant decrease in invasion occurring in the cells after a set amount of hours. So we know that the cell's movement has changed with this knockdown, but we wanted to see what the cell itself does with a lack of this IL-32 gene. Next, we performed scanning electron microscopy in order to see what the cells are looking like with and without this IL-32. So as you can see with the control, the cells go from a spindle-like structure that is aiding in their migratory and invasive properties to almost a normal-like stagnant cell that does not have these spindle-like structures causing it to be able to move and migrate and invade. So almost having a more stationary cell. We also wanted to see that we know since these extracellular matrix proteins are being made, are they actually being secreted and being used? So we used confocal imaging and we stained for fibronectin. And what we found was there was absolutely none in just a normal cancer stem cell rich cell line. However, when we knock down this IL-32, you have a drastic increase in the amount of fibronectin. So we're able to see that there's this intrinsic effect happening in the cell because of this IL-32, and there's an extracellular response that's occurring. And we see that there's a cell shape change and an environmental change for the cell. Next, we needed to see what is this protein that is having such a large impact on the properties of this cell. What does it look like? So we found that alpha and beta are the main isoforms that are being produced from this this protein. And from previous ELISA results, we know that this protein is not being secreted. So we have it's having a large effect intracellularly. So we wanted to see what it is doing intracellular. And this is why we looked at the structure of the protein itself. So we found that through previous literature, it's shown that the structure of IL-32 protein mimics that of a focal adhesion targeting domain. This is because of its very similar alpha helical bundle, as you can see, especially from the right side of the IL-32 structure, as well as what is not shown in RGD domain, or motif, excuse me. Because of this structure and the motif it has, its focal adhesion kinases are able to act and localize on these sites that IL-32 and fat domains are. Thus, you're almost doubling the function and activity of focal adhesions by having an increase in IL-32 on top of already having an increase in focal adhesion targeting domains because of the nature of these migratory and invasive cells already. So in conclusion, when you have a mammary cell that undergoes oncogenic transformation, specifically having these p53 mutations and an PI3K AKT activation, which would be caused by a delete of the P10 gene, you have this breast cancer stem cell. And this breast cancer stem cell has specific DNA methylation signatures that are causing IL-32 beta overexpression, which is ultimately leading to an extracellular matrix remodeling and cell invasion, causing an increase in the aggression of these breast cancer cases. So I would like to acknowledge and thank everyone that has been a part of this project for many years running, all working with Dr. Scholl in the biology department. And I also could not have done any of this work with previous studies done at the university, or at Augusta University with Dr. Korkaya, as well as Dr. Utsanova, who's at the Cancer Research Center of Lyon, and of course, South Carolina Embry program for funding everything. Thank you.